Dogs and wolves can look almost the same at a glance. Same eyes, same alert stance, but their DNA tells a different story. In January 2013, Nature published proof that dogs carry a genetic signature wolves never developed. Changes built for digesting starch, the food that came from living near people. That single shift helps explain how the wolf became the dog. If dog genetics is the kind of mystery you want to keep digging into, this is the channel for that. Hit subscribe, because the chapters ahead go even deeper into what DNA has been quietly hiding. That mattered because it turned an old assumption into something measurable. For years, people had guessed that dogs adapted to human scraps, to grains, to the leftovers of settlement life. The study did not leave that idea floating in folklore. It sequenced dog and wolf genomes, found 3.8 million genetic variants, and highlighted 36 regions of the genome as possible targets of selection during domestication. Among those regions, the researchers identified candidate genes and then added a functional clue that made the story sharper. Dogs showed evidence of greater starch digestion than wolves. The timing of the paper was important too, because the origin of the dog was still unsettled. The exact moment and place of domestication remained debated, and the genetic changes behind the shift from wolf to domestic dog were still poorly understood. That uncertainty is part of what gives the finding its force. It did not arrive as a tidy legend with a single answer. It arrived as a trace in the genome, a set of inherited marks showing that the dog's body had been pressed by a new world of food, one built around proximity to people. And that is what makes the result feel stranger the longer you sit with it. A dog did not simply learn to live near humans. Over time, some dogs seem to have carried a biochemical advantage for living near human meals, especially meals that included plant starch. In other words, the domesticated dog was not only changing in temperament or shape, its digestion was being rewritten by selection, one generation after another, until the animal's own DNA began to preserve the story of that shift. The study did not claim that every detail was settled, and that restraint is exactly what makes it credible. It showed a pattern strong enough to connect domestication with diet, but not so neat that the wider history disappeared. Instead, it opened a more unsettling question. If the dog's genome could remember what humans fed it, what else in domestication was being stored there in silence? For the first time, the answer was not a guess. It was written into the sequence. The trail tightens when the question stops being broad and becomes molecular. One gene comes into focus, AMY2B, the segment of DNA tied to pancreatic amylase, the enzyme that begins breaking starch apart before the food moves any farther through the body. That matters, because it turns a historical pattern into something measurable. Instead of saying dogs simply adapted to human food, the evidence points to a specific genetic change that helps explain how. This gene, AMY2B, turns out to be only one thread in a much larger genetic story, and it's exactly the kind of detail we unpack further in the complete material, where the hidden mutations behind dozens of breeds are laid out in full, one by one. You'll find that in the description and in the first comment. Stay with this, because the next piece of evidence complicates everything you just heard. In 2014, geneticist Maya Louise Arendt and her team at Uppsala University's Science for Life laboratory, publishing in Animal Genetics, connected AMY2B directly to a stronger capacity for starch digestion. The link was not vague or poetic. Amylase activity in dogs tracked with the number of AMY2B copies they carried. More copies, more enzyme activity. Fewer copies, less of that digestive push. It is a small shift in the code, but it changes the way the body handles a major nutrient. And then the picture gets more uneven. The variation was not spread evenly across the species like a universal badge of domestication. The same 2014 research observed copy number differences across 20 dog breeds, with patterns that were strongly breed-dependent. That means a Labrador, a Husky, a Shepherd, and a Terrier can all belong to the same domesticated species and still carry different versions of this digestive advantage. The adaptation is real, but it is not identical everywhere. That detail matters, because it challenges the simpler idea that domestication solved everything in one sweep. Instead, it suggests a more layered process, where some lineages kept getting pushed harder by diet than others. A breed living close to grain-rich scraps may have been under a different pressure than one shaped by leaner or more irregular food. Over time, Those differences could be read back from the copy number itself, almost like a tally mark left by survival. Seen that way, AMY2B becomes more than a gene name. It becomes a record of unequal access to the same human world. Not every dog inherited the same digestive machinery, and that unevenness is part of the evidence. The body kept count. The copies changed. The enzyme responded. And hidden inside that variation is the uncomfortable clarity of evolution at work. Some animals were better prepared to profit from what people left behind. This is where the story stops feeling like a single discovery and starts feeling like a decision point in history. A line was crossed somewhere between scavenging and living beside people, and the genome preserved the consequence. If you want to understand why dogs thrived so closely around human settlements, this is one of the clearest places to look. A gene, a copy number, an enzyme, and a set of breeds that no longer tell the same metabolic story. A sample can look decisive until a second paper places it under a harsher light. In 2014, a study led by Adam Friedman and Robert Wayne, Genome Sequencing, highlights the dynamic early history of dogs, published in PLOS Genetics, shifted the focus away from a single clean origin story. Instead of treating amylase expansion as a uniform leap that happened once and spread neatly through every lineage, the paper pointed to pre-existing variation in wolves and to little or no increase in dingo and husky lines. That detail mattered because it suggested the trait may not have arrived in one simple wave. Some animals already carried a different starting point before domestication tightened its grip. That is where the timeline begins to split. If wolves already held variation in amylase copies, then the advantage seen in farm-adjacent dogs might have been built from older raw material in some branches. But another reading appeared in 2016, when Maja-Louise Arendt and colleagues, publishing in the journal Heredity, proposed something sharper. Selection for efficient starch digestion may have acted on a new mutation in dogs, not only on variation inherited from an ancestral population shared with wolves. The tension between those ideas does not cancel the adaptation. It makes it harder, older in one sense, and younger in another, as if the same biological gain had different roots into different populations, and the evidence did not stay locked inside one narrow comparison. In 2015, a study by Seiya Tonoike and colleagues, published in Animal Genetics on Japanese native dog breeds, added another layer by confirming that increases in AMY2B copy number had already been suggested as a crucial genetic change during domestication. That matters because it shows the hypothesis did not remain isolated to one set of samples or one laboratory's interpretation. It kept resurfacing across breeds, regions, and methods, including the Japanese native lines, where AMY2B still carried the weight of an early domestication signal. So the question becomes less about whether dogs adapted to starch, and more about how many evolutionary paths could have led there. In one population, standing variation may have been enough to give certain animals a head start. In another, a newer mutation may have been pulled forward by the pressure of human food. The result is not a contradiction to be erased. It is a map of transition, showing that domestication did not move with one pulse, one date, or one mechanism. That kind of contradiction is exactly why so many dog owners end up going down this rabbit hole, because once you see how tangled the evolutionary trail really is, you start wanting more of it. The complete material walks through breed-by-breed comparisons, the ancient DNA timeline, a breakdown of which mutations came before domestication and which came after, and a plain language guide to reading a dog's genetic history. You'll find the link in the description and in the first comment. You can almost picture it in fragments. Wolves with an uneven genetic starting line, dogs living closer to grain-rich scraps, and researchers returning years later to ask why the metabolic footprint looks consistent while the origin story keeps changing. The answer is not neat, but it is more revealing. Selection can preserve different solutions to the same survival problem, and dog DNA seems to carry more than one of them. That is why this chapter matters. It turns a celebrated adaptation into a historical dispute with real biological stakes. Was starch digestion unlocked by ancient variation waiting in the background, or by a new change that gave certain dogs a fresh edge? The present answer is provisional but not vague. It says the domesticated genome may have stitched together both inheritance and innovation, leaving behind a record that is less linear than first assumed, and more faithful to the messy way evolution actually works. The first thing the studies do is pull the timeline out from under the simple story. That 2014 study by Friedman and colleagues did more than compare dog DNA with wolf DNA. It reached into the earliest chapters of domestication and came away with a sharper, less comfortable conclusion. The first dogs most likely appeared beside hunter-gatherers, not necessarily beside farmers. That detail changes the scene completely. It means the move toward life with humans did not have to wait for fields, grain stores, or settled agriculture. The pressure could already have been there in camps, along hunting routes, near shared scraps, in the long and uneven contact between people and animals living close enough to shape each other. And once that door opens, the starch question looks less like a neat agricultural invention and more like one thread in a much older weave. The 2013 Nature study had already hinted at that broader picture. It listed genomic regions under selection and pointed to genes with metabolic relevance, suggesting that domestication was never a single change written into one place in the genome. It was a spread of adjustments, each one carrying its own function, each one revealing a different way dogs were being pulled away from the wolf line. Seen from that angle, the ability to handle starch is not an isolated trick. It sits inside a larger reorganization of inheritance, one that involved feeding, movement, social contact, and survival in environments humans were steadily transforming. That is why the newer reading feels so important. It breaks the expectation that a carbohydrate-rich diet must have arrived first with agriculture and then forced the dog to adapt. Instead, the evidence points to a more fluid beginning. A dog could have lived close enough to people to benefit from human leftovers long before villages became permanent and crops became dominant. A human fire, a butchered carcass, a discarded tuber, a bowl emptied into the dirt. Any of these could have made the difference between a marginal advantage and a lasting trait. So the mutation story becomes less about a single switch and more about timing. If the earliest domestic dogs were already moving with hunter-gatherers, then the genes tied to digestion were being filtered by human behavior earlier than a farming model would suggest. The animal was not waiting at the edge of agriculture. It was already inside a social world where food habits changed from place to place, season to season, generation to generation. That kind of setting does something subtle to evolution. It rewards flexibility before it rewards specialization. You can almost see the consequence in the genome itself. Not a clean break, not one decisive moment, but a record of repeated contact with human food and repeated survival under pressure. The genetic marks do not say, "Here is the day dogs became domestic." They say something more complicated and more human. The relationship was already underway while the rules were still forming. If this is the kind of history that changes how you look at the dog sitting next to you, that's worth a like. And if there's a breed you want the genetics explained for, drop it in the comments. Subscribe too, while you're at it. Some of those requests end up becoming full episodes here. And that is where the deeper turn begins. The starch-related changes were not just evidence that dogs learned to live on scraps from human tables. They also show that domestication was never locked to a single economy. It could begin among mobile groups, adapt through mixed diets, and keep changing as human societies changed. The genome does not give a tidy origin. It gives a layered one, with hunter-gatherers, early contact, and metabolic selection all pressing on the same history at once. That leaves the adaptation looking richer than the old explanation allowed. Dogs did not simply become capable of digesting a human diet after agriculture appeared. Their biology was being negotiated in a world already shared with people, where survival depended on noticing the smallest advantage and keeping it. In that sense, the mutation is not the end of the story. It is the clue that the story began earlier, in a relationship so close that even food left behind by humans could help rewrite what a dog's lineage would become. By 2016, the picture had sharpened in a way that changed the whole conversation. The evidence no longer pointed only to an ancient shift during domestication. It showed that selection was still working, long after the earliest dogs had settled beside human lives. Researchers Taylor Reiter, Evelyn Jagoda, and Terence Capellini at Harvard University, publishing their study Dietary Variation and Evolution of Gene Copy Number Among Dog Breeds, found that positive selection continued to act on amylase copy number in breeds that lived on starch-rich diets. That detail matters because it separates one long process into two distinct chapters: the pressure that shaped dogs during domestication and the later pressure that refined specific breeds as their meals changed again. The result is not a neat ending. It is a deeper one, A dog's genome did not simply receive one useful change and stop there. It kept responding, generation after generation, to whatever humans placed in the bowl. Some breeds carried the marks of that history more strongly than others, which means the biology of feeding was being tuned in different directions depending on environment, selection, and time. The story became less about a single leap and more about repeated adjustments, each one leaving a trace. And then there was the claim that could have turned the whole finding into something darker. That same 2014 animal genetics study also tested whether AMY2B was linked to diabetes mellitus. The expectation was tempting, almost too tempting, because any gene tied to carbohydrate handling can sound like a warning sign. But in that dataset, no association was found. That absence is important. It keeps the science honest. More copies of a starch-related gene did not by themselves prove a path to diabetes. They proved adaptation, not destiny. That kind of discovery is why this channel exists, to keep tracing the DNA back to the moments that actually shaped it. If it changed how you see your own dog, subscribe, and pass it along to someone who'd want to know their dog's body has been keeping score too. So the final image is more precise than the popular version ever allowed. The dog did not carry one simple mutation that explained everything. It carried a record of changing diets, changing pressures, and changing human societies. That same 2016 heredity paper described diet-rich adaptations, including a significant gain in AMY2B copies, as a crucial step in the evolution of the dog from the wolf. Crucial, yes. Final, no. The genome shows a living archive of revision, not a single frozen moment. That is what makes the conclusion so powerful. The history of starch handling in dogs is not a shortcut to a headline. It is a timeline written in DNA, from the first domestication phase to later breed-specific selection, with no simple equation linking amylase and disease. What survives is something more revealing, a species shaped by proximity, by food, and by time, until even digestion became part of the bond. And if you want the real lesson hidden inside that bond, it is this. The body keeps score, but it also keeps adapting.